But this one, okay, and this one only goes to the simulcast to the other room.
Yeah,
right.
It's to the other room.
This is the on-off.
Turn, yeah.
Okay, do you hear me? Am I being heard? ¿Subimos? Okay, good morning. First, I'd like to announce, I was asked by the organizers to mention to you that at 5 p.m. today, there's going to be a panel on industry and academic approaches to deep learning research. And I'm happy to open the morning session, the third session of the meeting on inference, reasoning, and memory. The first speaker is Anna Schapiro from Harvard Medical School. Please.
Okay. Hi. Thank you so much for including me in this really fantastic conference. I've been enjoying it so much. Okay, so I want to start with the observation that we need our memory to serve two very different kinds of functions. We need to be able to remember individual experiences that we have, but we also need to be able to track the statistics and the relationships across experiences. So the first time that you go to a zoo and you encounter a penguin and a puffin, you want to store the memory that you saw this bird swimming but never flying, whereas you saw this very similar looking bird flying. So you want to resist generalizing across these experiences based on the fact that these animals look similar in order to access each of those individual memories. But on the other hand, you also want to be able to track the statistics across experiences in order to remember the fact, for example, that most things with feathers can fly. So in this case, you want to allow these memories to make contact with each other in order to see those relationships, whereas in this case, you want to keep the memories separate to avoid interference. So we have these two memory functions that are in tension. And one influential proposal for how the brain can support both of these kinds of memory is the complementary learning systems theory. Andrew introduced this a little bit yesterday, if you read his talk. But this theory says that the brain basically solves these competing problems by separating them anatomically. So there's a division of labor between the hippocampus and the neocortex, where the hippocampus specializes in the rapid learning of the specifics of individual experiences, and it will represent these experiences even if they're very similar to each other with separate neural populations, which allows you to avoid interference and recall each of those individual experiences. The hippocampus is then thought to replay these experiences offline, maybe especially during sleep, and this replay is thought to help the cortex to slowly extract the statistics across these experiences over long periods of time, like over days and weeks and months. And the neocortex is thought to use a different kind of neural code. It has these more overlapping representations that are useful for generalizing across experiences. So the hippocampus would represent these experiences of seeing a penguin swim and a puffin fly with these separate neural populations, whereas the neocortex would represent all these memories of seeing birds fly with this more overlapping code. And that's useful because if you see a new animal with feathers, you'll immediately be able to see its relationship to all these other animals with feathers, and you'll be able to infer that it has similar properties, that it can probably also fly, for example. Okay, so that's all good. This framework has been, I think, very useful in helping us understand how the hippocampus and the cortex collaborate in memory. But there are some really important gaps, and some of my research has focused on trying to fill in these gaps. One gap is that it doesn't explain how we can pick up on novel regularities in our environment quickly on a short time scale, so like over a few minutes or hours, which is something that we're very good at, but it doesn't fit because we have the story about kind of rapid episodic learning and then slow statistical learning, but that just leaves out this ability to rapidly pick up on statistics in the environment. I'm going to try to convince you that actually the hippocampus is capable of doing that in addition to its role in episodic memory. And then another question is what happens after this information is encoded? So how is this information consolidated? How does the hippocampus teach this new kind of information about regularities to cortical areas offline? What does that process look like? My approach to these questions is to use a combination of computational modeling, neuroimaging, and behavioral work. I think this is a useful way to bridge from the brain to the cognitive and behavioral phenomena that it supports. So you'll see that the computational modeling approach that I take is low level enough to make direct contact with neural representations as measured with neuroimaging or with physiology, but also high level enough to perform tasks that are closely analogous to the ones that we're using in our human behavioral experiments. So the models provide theories and mechanistic accounts that are informed by neural and behavioral data, and then they make predictions that we can test in both kinds of experiments. So let's apply this approach to this first question: how do we learn novel regularities? So I'll show you some empirical evidence from fMRI and patient studies that suggests that there's a role for the hippocampus in this kind of learning. And then I'll show you a computational model that tries to address this tension of one area supporting both of these functions. These are computationally in tension. How is it possible that the hippocampus is doing both of these things? I'll show you a neural network model that tries to address that. Okay, so let's start with the empirical evidence. So I've been using temporal statistical learning paradigms where participants see a stream of novel images presented one at a time. It might look like this. Participants have some kind of cover task. In this particular experiment, they were looking for patches in the image that had been turned to grayscale. So it's just a orthogonal cover task, but unbeknownst to the participants, there are these hidden regularities in the sequences. So in this first experiment, I'll show you there were pairs of items that always occurred together in the sequence. So two items from a pair
are seen much more frequently back to back than two items that might occur together in the transition from one pair to the next. We wanted to test whether the hippocampus might be sensitive to these kinds of regularities. And to do this, we exposed participants to 40 minutes of these sequences with the pair structure. And before and after that exposure, we measured the representations of each of the individual items in the fMRI scanner in the hippocampus. So we present each image, and we look at the pattern of activity that's evoked across all of the voxels in the hippocampus, and then we record that pattern. And then we can ask, are two items that were paired in the sequence represented more similarly than two items that were not paired in the sequence? And what we find is indeed that over the course of this learning, the hippocampus comes to represent these items that were paired as more similar relative to items that were not paired in the sequence. And the hippocampus seems to be using this kind of overlapping neural code here that's maybe not what we're used to seeing in the hippocampus. So we're going to come back to this. So we have some evidence that the hippocampus is involved in this kind of statistical learning, but we wanted to get at whether it's really the key region necessary for this learning. And so we tested a patient with complete bilateral hippocampal damage to see what would happen if she would be able to learn these kinds of sequences. So we exposed her to the same kind of pair structure. And afterwards, we asked her, which feels more familiar to you? These two items that came from a pair that she had seen regularly in the sequence, or these two items that she had seen but were not paired together in the sequence. And we did this for these shape stimuli and then also for syllables and scenes and tones to see if this is something kind of common across modalities and stimulus types. And we found across all of these different tasks that she performed much worse than controls and never above chance. And there's been now a replication of this with 4 additional patients. So we have some confidence that the hippocampus is really necessary for this kind of learning. Exactly. So that was our control experiment. Yes. So we had another condition where we just asked her, do you even recognize this item relative to a new item that you haven't seen? And she was able to do that. Yeah, exactly. So we saw that the hippocampus was involved in learning these pairs of items that occurred frequently together in the sequence, but we think that real-world temporal structure is much richer than that. And we wanted to test whether the hippocampus might be sensitive to higher-level forms of structure. So in this experiment, we exposed participants again to a sequence of images, just one at a time. But in this case, the sequence was generated from a random walk on this graph with community structure. And this graph has strong temporal structure, but all subsequences in this graph are seen equally frequently. So you can't use the transition probability as a cue in this case. You have to be sensitive to kind of the higher level structure here. And we found that the hippocampus was sensitive in the same way to this kind of structure. So items within a community are represented more similarly than items across a community. So it seems like the hippocampus can learn this more kind of realistic, sophisticated form of structure as well. Okay, so how do we learn novel regularities? I showed you this evidence from the pair structure and community structure that the hippocampus has these representations that are coming to reflect the temporal structure of the environment. And I showed you a patient study that suggests that the hippocampus might be necessary for this kind of learning. Okay, so now I want to ask the question, how does this actually happen? What's the mechanism for this? In particular, we have to address this tension of one area being responsible for remembering both statistics and episodes. So how do we reconcile evidence of the hippocampus integrating statistics across experiences and coming to represent associated items similarly with what we know about the hippocampus specializing in keeping experiences separate to avoid interference? These are very different kinds of representations. How is it that the hippocampus is involved in both of these things? So to try to address this, we took a model of the hippocampus that we know is very good at episodic memory, and we exposed it to sequences with statistical structure to see what would happen if it would be able to learn them. So this is a model that has connectivity and properties that reflect what we know about the actual physiology of the hippocampus. We have a layer that represents superficial layers of entorhinal cortex, that's the input to the hippocampus. We have 3 hidden layers that represent 3 subfields of the hippocampus, dentate gyrus, CA3 and CA1, and then we have deep layers of ventral cortex representing the output from the hippocampus. So this model is an autoencoder. It's just trying to reproduce whatever it gets in this input layer on the output layers. It's learning whatever weights it needs to learn to do that mapping. In case you haven't seen this particular kind of diagram before, the height and the color of the squares here represents the firing rate of a neuron or a small population of neurons, and then these arrows represent connectivity. The connectivity actually has different properties throughout this network. It looks kind of like this if you were looking at all of the connections, but of course that's a mess, so I'm just going to leave it with these big arrows. So there are 2 pathways through this network and in the actual hippocampus. There's the trisynaptic pathway and then this monosynaptic pathway. And these 2 pathways have different properties. So the trisynaptic pathway is really the pathway that implements these sparse pattern-separated representations that are the heart of the reason that we think that the hippocampus is really good at episodic memory. And it has these kinds of representations because there's very sparse connectivity amongst the layers in this pathway. There's also high inhibition within the layers. So that means that 2 inputs coming in, even if they're very similar to each other, are going to get projected to different patterns, different sets of units in these layers. The monosynaptic pathway, which has region CA1 here, has different properties. It seems to have a more intermediate amount of overlap, and that's because it has more connectivity with the regions that it's connected to. And in previous episodic memory simulations with this model, this pathway has been important for translating between these really sparse representations in the trisynaptic pathway and then distributed representations out in cortex. So this kind of intermediate amount of overlap is useful for providing that translation function. One last property I want to point out about these 2 pathways, the trisynaptic pathway seems to have a faster learning rate than the monosynaptic pathway. So given a particular learning experience, the trisynaptic pathway will make a bigger change in its weights than the monosynaptic pathway. Okay. Yes? Yes?
So when you're in CA1, is it the same neurons that are involved in most representation, or except for units in CA1 that are involved in other representations?
They are overlapping, much more overlapping. So I'll actually show you in a minute a simulation. Am I going to have to go through all these animations? I'll show you a simulation where you can see different inputs coming in. You'll be able to see that there's much more overlap across the different inputs coming in CA1 than there is in the trisynaptic pathway. Almost there. I'll show you that in a minute. Okay, so first let me show you the way that I present the sequential structure to this model. So I'm using a very simple kind of input where I present the current item with full activity and then the previous item with some decayed activity. So it only has access to 2 items at a time, and then I just move that window along, and the model has to learn from the statistics over time that there are these pairs that will keep occurring together in the sequence. Okay, so here's what that looks like in the actual model. Here I'm presenting the second item in a pair, where the first item in the pair has some decayed activity. And then we can move on to the next item. So here's the first item of a new pair, and this item that we were just on has now kind of decayed away. So here's what it looks like when the model is training, and you can see that dentate and CA3 have these very sparse representations, and each different input that's coming in is producing a totally different pattern in those regions, whereas CA1 has a higher proportion of activity overall, and you'll see that some of those same units keep coming up across the different inputs. So that's the difference in the overlap between this pathway and this pathway. And then we'll contrast that statistical learning kind of scenario with an episodic variant of the task, where we just present the pairs directly to the model without the transitions between the pairs. And this is really just a standard episodic memory simulation. So if I tell you that these 2 things go together, memorize that they go together. And we'll see how the model kind of does on this task relative to the one where you have to extract the structure over time. So after the training is finished, we want to look on our test trials with this model and do something that's
analogous to what we were doing with the fMRI pattern analysis approach. So that's very easy to do in these models. We present items by themselves at test, and we just measure the pattern of activity evoked across all these different hidden layers. We record that, so there's the pattern of activity for item A and the pattern of activity for item B. So we collect all of those patterns, and then we can do the same analysis that we did before. We can ask, are items that were paired in the sequence represented more similarly than items that were not paired in the sequence? So this is just to orient you to the kind of plot I'll show you in a second. So first, I'll show you the results for the episodic variant of the task, where the pairs were clearly demarcated for the model. So again, this is going to be the similarity of the representation of each item to each other item. And what we see in the trisynaptic pathway, so in dentate and CA3, is that there's this really nice, strong pair structure. So items A and B that were paired together are represented basically the same in these two regions, whereas CA1 only weakly represents this structure. So this tells us what we already knew really about this model, which is that the trisynaptic pathway is very good at learning these clearly demarcated episodes quickly. So now what happens in the case where the model has to extract these pairs from the statistics over time? Here we see a totally different pattern of these representations. So now dentate and CA3 are not showing any sensitivity to the pair structure at all, whereas CA1 is now showing sensitivity to the structure. And this checkerboard structure is interesting. It suggests that the trisynaptic pathway is trying to memorize both the pairs and the transitions between the pairs. So a transition like BG is represented just as, or items B and G from a transition are represented just as similarly as two items from an actual pair, as opposed to two items like A and G that were never seen together, which are not represented similarly. So, so this pathway is doing too good a job of trying to memorize everything that it sees, which is useful when you're trying to remember the details of your experience for episodic memory, but it's not useful for this learning problem where you're trying to extract the structure over time. So it looks like these different layers have different representations depending on the learning problem and the structure of the input. And CA1 seems to be responsible for this particular kind of learning. So it could be that this is underlying what we're seeing in the fMRI data where we saw those kind of merging of representations. And it turns out that this model can handle that community structure case as well. So it can account for that data too. So what's happening here? Why is it that the monosynaptic pathway is doing such a good job at statistical learning? Well, I had pointed out these properties that the trisynaptic pathway has this fast learning rate, separated representations. The monosynaptic pathway has a slower learning rate, more overlapping representations. If you remember from the beginning of the talk, these are exactly the properties that separate the hippocampus and the cortex in the complementary learning systems theory. So the hippocampus does fast learning on separated representations, and the cortex does slow learning on overlapping representations. So the monosynaptic pathway is good at statistical learning for the same reasons that cortex is good at statistical learning, which is that slow learning helps you to integrate information over long periods of time, and overlapping representations help to highlight commonalities across experiences, basically. So it's like having a little piece of cortex within the hippocampus, although we think that it's not as overlapping and not as slow learning as the cortex. So, so how is it that one area is supporting both episodic memory and statistical learning? Well, the hippocampus is not homogeneous, and we're proposing that it's really the trisynaptic pathway that's responsible for keeping episodes separate to avoid interference, but that this other pathway actually has properties that are very well suited to this rapid statistical learning, so we think the monosynaptic pathway might be doing that. So I started with this idea that the hippocampus and the cortex are complementary learning systems, but we think that there might be complementary learning systems within the hippocampus as well that are kind of a microcosm of this larger system, where we think it's still the case that the cortex is extracting statistics over even longer periods of time using an even more overlapping kind of representation. Okay, so now I want to move to my second question, which is how does this transformation happen? So how do we think this systems consolidation process might be happening from the hippocampus to the neocortex? So how is new structured information consolidated? I'm going to go over some empirical evidence for how we think this process might happen, especially we think sleep is a especially important period for this consolidation, so I'll talk about some work looking at the role of sleep. And then again, I'll show you a neural network model that tries to address the mechanism of how the hippocampus could actually be teaching information to cortex offline. So first, why do we sleep? We spend an enormous amount of our time sleeping, and it would be nice to understand why we do this. There's probably a lot of reasons that we sleep, but I think that one important reason is that it gives us a chance to kind of disconnect from our environment and process information without having to encode anything new. So during the day, you are bombarded with sensory experience that you're trying to process and encode, and then at night you have a chance to try to update your internal models of the world without any further need to process that external environment. And we think that hippocampal replay during sleep is an important part of this process of kind of reflecting on what happened during the day and helping to establish this new information in cortical networks. So how is new structured information consolidated during sleep? This is a very rich and complicated question, and I'll tell you about kind of two of the more specific questions that we've been asking to start to get at this. One basic question is just how do we even decide what to process during offline periods? So it's probably not possible to replay every event that you experience during the day, and even if you could do that, it's probably not worthwhile to replay every single experience you've had. So how do we decide what to replay? So maybe you only got a glimpse of the penguin at the zoo, but you spent a lot of time with the puffin. Is sleep more interested in trying to process that penguin memory, which is maybe weaker and needs more help, or is it more interested in the puffin memory, which is maybe more salient or strong? We don't know how sleep might prioritize those kinds of memories. Another question is, what are the consequences of this transformation for memory? So one prediction of this account is that as memories come to rely more on a cortical neural substrate, that you'd expect a benefit in memory for commonalities across experiences, since this kind of overlapping code should highlight those commonalities for you. So you might expect a benefit in memory for commonalities across birds that you've seen, as opposed to their kind of individuating idiosyncratic features. So the paradigm that we've been using to try to address these questions is an object category learning paradigm, where participants learn about these three categories of satellite objects. So satellites in the same category share most of their features. A particular satellite will have all of these kind of parts that are shared with other members of its category. It also has a class name that participants learn. But each satellite also has unique individuating features, so a part that no other satellite has, and then a unique code name. And we teach people about these objects by showing them a satellite with a missing part, and they have to do their best to try to fill in that part. They have this like point and click GUI where they fill in a missing part, and then we give them feedback about whether they've done that correctly or not, and they just keep going with that until they reach some learning criterion. The unique features of the individual satellites are much more difficult to remember than the features that are shared across satellites from the same category. And so we query people on the unique features much more often, so that by the end of training, their memory for unique and shared features is matched. And that's important because we want to look at how those two things change across a period of sleep. And so we want to make sure that they're equal by the end of training. And then to test people, we again just show them a satellite with a missing part. They do their best to fill it in. And then at test, we don't give them any feedback about whether they've done that correctly. So to look at how memory changes across sleep relative to a day awake for this kind of memory, we have a kind of a standard sleep design where we have a sleep group that learns about the satellites in the evening, they have an immediate test, and then they come back in the morning for a delayed test. And then we have a wake group that does their learning in the morning, an immediate test, and then they come back in the evening for a delayed test. So here I'll show you the change in proportion correct from the first to the second session, separately for the unique features and the shared features, and then separately for the sleep and wake group. So what we see is that there's no change in memory for unique features across a night of sleep. You're able to retain that kind of information, whereas there's a lot of forgetting of those kinds of features across a day awake. So this is kind of standard, like what we typically see in the sleep literature. Sleep prevents the deterioration of these unique features. But the condition that hadn't really been done before is this shared feature. So what happens for this information that has these commonalities across the category members? And what we see is that there's actually an enhancement of this memory across a night of sleep, whereas again, there's forgetting across a day awake. So sleep somehow seems to promote a better understanding of the shared structure of these category members. And we think that's consistent with this idea that if memories are starting to come to rely more on this neocortical substrate, that they'll benefit from this overlapping representation that highlights the commonalities across experiences for you. We also ran an fMRI variant of the same paradigm to try to get more directly at the role of hippocampal replay in this consolidation process, and also to look at how memories are prioritized for that replay, that kind of offline processing. So here, it's a similar design. We have a sleep and a wake group. We have training either in the evening or in the morning, an immediate test. And then people get into the scanner and we measure the representations of each of the individual satellites the same way that I showed you earlier. And then we have an awake rest period where we're going to try to look for hippocampal replay. So we're not yet looking at hippocampal replay during sleep. In this experiment, we're just looking at awake hippocampal replay. And then we bring people back. We have another awake rest period to look for replay, and then a final memory test. Right, okay. So what we find in this experiment is that in the second session, where we have this rest period that comes right before a memory test, we find that there is a positive correlation between the amount of replay that we detect in the hippocampus and memory for particular satellites. So if we detect that a particular satellite has been replayed a lot during this rest period, we're able to predict that that satellite will improve in memory, or that satellite will have better memory. So that's what these positive correlations mean in second session. So that's exciting. It suggests that we're able to detect hippocampal replay in humans and that it relates to memory. But it doesn't necessarily suggest a causal link between this replay and the memory. It could be that people are just replaying things that they already know better. It's not that that replay is helping the memory. But when we look in the first session, where we have a test that happens before the replay period, we actually find negative correlations. So what this means is that if you have poor memory of a satellite during during this test, then you will have more replay of that satellite in the subsequent rest period. So that's not consistent with a story where you're just replaying things more that you know better. It suggests that the hippocampus is doing something adaptive, where it's replaying the things that need help. It's replaying the things that were weaker, but that the things that it replays are going to be improved. So that's, it suggests the hippocampus is really doing something smart and adaptive. We can also look at what happens, so how does this awake hippocampal replay relate to changes in memory from the first to the second session, and now we're going to separate it out into those two groups that had different experiences between the two sessions. And we find that this awake hippocampal replay that we're measuring is only related to memory improvement for the group that slept in between the two sessions. So that suggests that the replay that we're measuring is either influencing what happens during the night and in the rest of that intervening period, or maybe more likely it's just representative of what continues to happen in the intervening period, and that replay that happens during the night is especially useful for this kind of memory consolidation. Okay, so
I've shown you that sleep enhances memory for regularities while preserving unique features, and that wake replay in the hippocampus seems to prioritize weaker memories, which are the memories that need more help, so we think that makes some sense. And it predicts memory improvement only over a period of sleep. So how is structured information consolidated? I showed you that sleep seems to adaptively stabilize memories, perhaps increasing reliance on cortical networks. And now I want to show you our attempt at trying to understand the mechanism for this consolidation process. So how is it that hippocampal replay might shape cortical representations? So here we have another neural network model that has interacting hippocampal and cortical components. So we teach this model about the satellite objects in the same way that we teach people about the satellites. We show it we show a satellite with a missing feature. It tries its best to make a guess about how to fill in that feature. We give it feedback. That's just really standard normal neural network learning, so that's all fine and good. So it's able to learn about the satellites during the day. Then the question is, what happens offline? How do we get this model to do something useful without any further input from the environment during sleep? So here's what we've come up with. So doing useful learning during sleep is a hard computational problem because, like I said, normally our models are relying on input and feedback from the environment. So what do we do? How do we do useful learning on our kind of existing internal representations without any further input from the environment? And here's sort of the key features that we've come up with here to get this to work. First, we just need a way of visiting different memories. So we want the model to be able to operate completely autonomously, and it has to be able to just move from memory to memory by itself. And here we're just using synaptic depression. So if there's a particular kind of attractor that the network is in, it can't stay there forever because two units that are coactive, the synapse between them will tire out over time, and then it'll have to move into a new memory. So it's just a way of getting the network to replay different memories. Then we need a way of marking which states of the network are worth learning about. And here we just use network stability. It's a very kind of simple measure we think the network could track to say this is a state that's worth learning about. We call it a plus state. So when the pattern of activity is very similar, like right now, from time point to time point, that will get marked as a state that's worth learning about. Then we need a way of revealing bad states of the network or contrast to these good states. And here we use oscillations, which are a very kind of prominent aspect of the sleeping brain, lots of different kinds of oscillations. We're going to use them as a way of perturbing these attractor states and revealing bad states of the network that are kind of bad in a useful way. So let me try to explain that. So we're oscillating the level of inhibition in the network, which just controls how much activity is possible. And when we raise inhibition above baseline levels, let's say we're replaying a memory of this satellite here, the weakest parts of this attractor will fall out first as you raise inhibition. So it's just a way of identifying weak parts of the memory that might need strengthening. So that's kind of one useful bad state that an oscillation will reveal. When you lower inhibition below baseline, that allows activity to spread farther than it normally would, and that can reveal potentially competing or interfering memories that are nearby in this kind of attractor space. So now we've identified memories that we might want to kind of push away from this memory in order to avoid interference. So it's another bad but usefully bad state of the network, and we record those states. And now we can do area-driven learning. So in this case, we're using contrastive Hebbian learning, which is just a biologically plausible form of local area-driven learning, where we adjust the connection weights so that the pattern of coactivity in these minus states and these bad states look more like the pattern of coactivity in these good plus stable states. So this is the way that we're able to get useful area-driven learning operating autonomously offline. So what do we find? we find that over the course of the model's sleep, the shared parts of the cortical representations of satellites from the same category are enhanced, which is what you'd expect. But we also find that the unique parts, the kind of individuating pieces of these individual satellites are also preserved. It's not showing up on this thing, but I've colored in these little pieces. So we're really excited about that because it corresponds really nicely to the behavioral data. So where we're seeing this, enhancement of shared features, but preservation of unique features, that's a very natural thing for this type of model to account for. So it looks like we're kind of on the right track in thinking about how these cortical representations might be changing as a function of hippocampal replay. This model makes a prediction that we've now tested. So I think this is the last experiment I'll show you. So one prediction of this model is that the hippocampus might be important for the replay of information during sleep, even if that information was not, even if the hippocampus was not necessary for encoding that information to start with. So it's a very counterintuitive prediction. The hippocampus is needed in this model to help kind of reinstate the context and the details of an experience, even when that experience didn't require the hippocampus for kind of initial encoding and behavior. So here we have tested this prediction in amnesic patients with hippocampal damage on this task called the motor sequence task. It's a very simple task. Participants just type a sequence like 4, 1, 3, 2, 4 over and over with their left hand. That's the whole task. So we don't expect that hippocampal amnesics will have any trouble with this task. But this task has a big sleep-dependent component to it, and we were curious whether these patients would show that sleep-dependent enhancement. And what we see is that the controls and the amnesics perform the same across 12 trials of initial training. That's what we expected. But they separate out 24 hours later after a night of sleep. And so this is exactly what we predicted. It looks like there's learning that you can do without the hippocampus that you won't be able to consolidate without the hippocampus. So the hippocampus might play a broader role in memory consolidation during sleep than just information that is dependent on the hippocampus at encoding. Okay, so I showed you this model that neural systems might be able to leverage oscillations during sleep to do this useful autonomous learning, and some of our experiments suggest that this model might be on the right track. So back to the big picture, I showed you that the hippocampus is critical for rapidly learning regularities, and we're arguing that it might, in particular, be relying on this monosynaptic pathway that I think has properties that are very well suited to this kind of learning. And then hippocampal replay and sleep help to situate these representations in long-term cortical stores. So together, I think this research is starting to give us some new insight into how patterns in our experience are initially encoded and then how they're transforming over time. Okay, thank you.